Understanding skeletal muscle contraction begins with understanding a few terminology which make up the skeletal muscle. So the skeletal muscle is actually made up of muscle fibers, which are also known as myocytes. Each myocyte is made up of myofibrils, and these myofibrils are made up of sarcomeres, and these sarcomeres, they are made up of thick filaments and thin filaments. The thick filaments are made up of a protein called myosin, and the thin filaments are made up of a protein called actin. They also have other proteins called tropomyosin and troponin. And troponin has three components: troponin I, troponin T, and troponin C. The sarcomere now, again, let us. Sarcomere, as we have already discussed, has thick filaments and thin filaments. The myosin interacts with the actin via the sliding filament model and the cross bridge contraction model to bring about skeletal muscle contraction. So one of the important terminology is sarcomere, which is the basic contractile unit of the skeletal muscle. Basic contractile unit of the skeletal muscle. There are three other terms that we need to understand, which is confusing. One is sarcolemma, the other is sarcoplasm, and another is sarcoplasmic reticulum. It's very easy to understand if we again go back to the anatomy. So the muscle, so we are repeating the same thing so that we better understand it. So the muscle is made up of myocyte. Myocyte is made up of myofibril. Myofibril is made up of sarcomere, and the sarcomeres have myosin and actin, which is myosin is the thick filament, and actin, troponin, and tropomyosin, they are the part of the thin filament. Now this myocyte is the basic cell of a muscle unit. So just like any cell, it has a cell membrane, it has cytoplasm, and it has an endoplasmic reticulum. If we understand this, the definitions become very easy. So this myocyte cell membrane, the myocyte cell membrane is known as the sarcolemma. The myocyte cytoplasm is the sarcoplasm, and the myocyte endoplasmic reticulum is the sarcoplasmic reticulum. So this is the basic anatomy. Again, the cell membrane of the myocyte, the cell membrane of the myocyte is sarcolemma. The cytoplasm of the myocyte is sarcoplasm, and the endoplasmic reticulum of the myocyte is sarcoplasmic reticulum. Now that we have understood the basic structural organization, let us come to myosin. So the myosin is what forms the thick filament of the sarcomere. It has 6 chains. 1 pair of heavy chains and 2 pairs of light chains. So total 6 chains. The heavy chains, they are arranged in a alpha helical manner like this, and the light chains, with the terminal end of the heavy chain, the 4 light chains are arranged like this, and they form the myosin head. So myosin head is formed by the light chains, and the myosin tail is formed by the heavy chains. Myosin head light chains, this myosin head, it acts as the actin binding site. It acts as the actin binding site, and we have to remember that this also has the myosin ATPase activity, which is required for contraction of skeletal muscle. In fact, the contraction of skeletal muscle depends upon two important things. One is ATP and another is acetylcholine. In fact, the ATP mediated contraction, it precedes the acetylcholine mediated contraction of the skeletal muscle. Hence that myosin head, it has an actin binding site and the myosin ATPase activity. Now, the actin plus tropomyosin plus troponin, they form the light chains. So, the actin is arranged like this. Now, I'll just pull up a picture, it's better. So, this is how the myosin heavy chain with light chain, and this is the actin binding site of myosin. So, the light filaments, they look like this. These blue balls are actin proteins, and we can see that the tropomyosin is wrapped around actin, and it forms the myosin binding sites of actin. When the muscle is not contracting, the myosin binding sites of actin are covered with tropomyosin. We can also see troponin protein over here, which is composed of troponin I, troponin C and troponin T. Troponin T helps to bind troponin to tropomyosin. Troponin I inhibits the binding of myosin to actin. Along with tropomyosin, troponin I inhibits the binding of myosin to actin. Troponin C is the calcium sensing part of troponin. Once the intracellular calcium level increases, the troponin C binds to the calcium. There is a conformational change in the troponin molecule, and this conformational change results in the removal of the tropomyosin from the actin, and so the myosin binding sites of the actin, they are freed, and now myosin can bind to actin and there can be contraction. Now let us see how a sarcomere looks microscopically. We can see that the sarcomere, there are two vertical blue lines. These are the Z discs, and the part of the diagram that lies between the two Z discs is what makes one sarcomere. If we draw a line in between the two Z lines, we find an M line. From the M line, we can see there are three thick filaments running, and these filaments are the myosin heavy chains. On the Z disc, we can see the blue filaments. These are thin filaments, and these are the actin filaments. Now, there are a few terminology which is important to remember. We can see that there is an area here where the area in this part where there are no thin filaments. There is only thick filament. This is known as the bare zone. And we can see a band which runs from here to here, here to here, a band which runs from here to here, where there is overlap of the thick filament and the thin filament, overlap of the thick filament and the thin filament. And this is the bare zone. We can see that there is no overlap of thick filament and thin filament over here. Just like a band where there is overlap of thick and thin filament, we can find something called I band. in this I band we only have thin filament. We can see that there are no thick filaments over here. So this I band corresponds to the bare zone. Bare zone only has thick filaments and the I band only has thin filaments. So this is basically the structure of a sarcomere, which is the basic contractile unit of proteins. Now these sarcomeres, they are not floating around in the myofibrils. They have certain proteins are there which anchor these myofibrils inside the cell. Otherwise they couldn't maintain this configuration. These proteins are called cytoskeletal proteins, and one of the important cytoskeletal protein that we know is dystrophin. Now dystrophin is a actin binding protein which binds to actin and helps to anchor this sarcomere to the cell. We know that a defective dystrophin is found in Duchenne muscular dystrophy. There is another kind of protein called titin. Titin, which is implicated in cardiomyopathies. Now this titin, it runs along the length of the thick filament and anchors the thick filament to the Z line. There is one more protein known as nebulin and another known as alpha actinin. This nebulin protein runs along the whole length of the thin filaments and gives its structural integrity, and alpha actinin anchors the thin filaments to the Z disc. So this is in summary the structural organization of a skeletal muscle, which is difficult to understand, but if we go through this video once or twice it will be easier. In the next video we shall be going into details of skeletal muscle contraction, which is very high yield in terms of our understanding and exams.